Hello there. Is there really a gene responsible for our religiosity? And what's the true foundation for the VMAT2, aka the God gene? Let's check it out in this video. And don't forget to support the valuable content of the Curious Tube by hitting subscribe, like, and notification icon.
Dean Hamer claims to have found the God gene, VMAT2 in humans. Dean Hamer is a geneticist and director of the Gene Structure and Regulation Unit at the National Cancer Institute in Bethesda, Maryland. After comparing more than 2,000 DNA samples, he has concluded that a person's capacity to believe in God is linked to brain chemicals. However, his conclusions have been criticized by some clerics and scientists. For example, John Polkinghorne says the idea of a God gene goes against all my personal theological convictions. You can't cut faith down to the lowest common denominator of genetic survival. It shows the poverty of reductionist thinking. Carl Zimmer claims that VMAT2 can be characterized as a gene that accounts for less than 1% of the variance of self-transcendence scores. These, Zimmer says, can signify anything from belonging to the Green Party to believing in ESP. The God gene hypothesis proposes that a specific gene, called vesicular monoamine transporter 2 VMAT2, predisposes humans towards spiritual or mystic experiences. The idea has been postulated by geneticist Dean Hamer, the director of the Gene Structure and Regulation Unit at the U.S. National Cancer Institute, and author of the 2005 book The God Gene: How Faith is Hardwired into Our Genes.
The God gene hypothesis is based on a combination of behavioral genetic, neurobiological, and psychological studies. The major arguments of the hypothesis are: spirituality can be quantified by psychometric measurements. The underlying tendency to spirituality is partially heritable. Part of this heritability can be attributed to the gene VMAT2. This gene acts by altering monoamine levels. And spiritual individuals are favored by natural selection because they are provided with an innate sense of optimism, the latter producing positive effects at either a physical or psychological level. According to this hypothesis, the God gene is a physiological arrangement that produces the sensations associated, by some, with mystic experiences, including the presence of God or others, or more specifically spirituality as a state of mind, based on research by psychologist Robert Cloninger. This tendency towards spirituality is quantified by the self-transcendence scale, which is composed of 3 subsets: self-forgetfulness, as in the tendency to become totally absorbed in some activity, such as reading, transpersonal identification, and mysticism, an openness to believe things that remain unproven, such as ESP. Cloninger suggests that taken together, these measurements are a reasonable way to quantify how spiritual someone is feeling. The self-transcendence measure was shown to be heritable by classical twin studies conducted by Lyndon Eves and Nicholas Martin. Interpreters of these studies argue that specific religious beliefs have no genetic basis and are instead memes, cultural units transmitted by imitation, in order to identify some of the specific genes involved in self-transcendence. Hamer analyzed DNA and personality score data from over 1,000 individuals and identified one particular locus, VMAT2, with a significant correlation. VMAT2 codes for a vesicular monoamine transporter that plays a key role in regulating the levels of the brain chemicals serotonin, dopamine, and norepinephrine. These monoamine transmitters are in turn postulated to play an important role in regulating the brain activities associated with mystic beliefs.
The evolutionary advantage this might convey, or whether it could be a side effect of a separate adaptation, have yet to be fully explored. However, Dr. Hamer has hypothesized that self-transcendence makes people more optimistic, which makes them healthier and likely to have more children. Although it is always difficult to determine the many interacting functions of a gene, VMAT2 appears to be involved in the transport of monoamine neurotransmitters across the synapses of the brain. P.Z. Myers argues, it's a pump, a teeny pump responsible for packaging a neurotransmitter for export during brain activity. Yes, it's important, and it may even be active and necessary during higher-order processing, like religious thought. But one thing it isn't is a God gene. Carl Zimmer claimed that VMAT2 could be characterized as a gene that accounts for less than 1% of the variance of self-transcendence scores. These, Zimmer says, can signify anything from belonging to the Green Party to believing in ESP. Zimmer also points out that the God gene theory is based on only one unpublished, unreplicated study. However, Hamer notes that the importance of the VMAT2 finding is not that it explains all spiritual or religious feelings, but rather that it points the way toward one neurobiological pathway that may be important. The vesicular monoamine transporter 2, also known as solute carrier family 18 member 2, is a protein that in humans is encoded by the SLC18A2 gene. VMAT2 is an integral membrane protein that transports monoamines, particularly neurotransmitters such as dopamine, norepinephrine, serotonin, and histamine from cellular cytosol into synaptic vesicles. In nigrostriatal pathway and mesolimbic pathway dopamine-releasing neurons, VMAT2 function is also necessary for the vesicular release of the neurotransmitter GABA.
Binding site is that of dihydrotribnosine, DTBZ, and reserpine. Globulin binds at this site. Amphetamine and methamphetamine bind at distinct sites on VMAT2 to inhibit its function, although the amphetamines inhibit VMAT2 presynaptically, leading to diminished neurotransmitter. The primary mechanism for the enhancement of extracellular monoamines, like dopamine, is reversal of the dopamine transporter. Other VMAT2 inhibitors such as GZ793A inhibit the reinforcing effects of methamphetamine, but without producing stimulant or reinforcing effects themselves. VMAT2 is essential in the presynaptic neuron's ability to facilitate the release of neurotransmitters into the synaptic cleft. If VMAT2 function is inhibited or compromised, neurotransmitters, such as dopamine, cannot be released via normal transport into the synapse.
Thanks for watching. Give this video a thumbs up if you want to see more videos from this channel. If you knew of any useful information on this topic, let me know in the comments below. And if you're new here, click on the subscribe button and notifications icon for more useful information and tips from The Curious Tube.